All right, hello everybody. So today I'll be walking through the process that came from the article Multipotentiality of Single Embryonal Carcinoma Cells by Kleinsmith and Pierce. So like I said earlier in the article, the main problem plaguing the experiment was the burden of proof that every single teratocarcinoma cell was pluripotent and not just certain of them. So I'll be explaining the process of how specifically they isolated the teratocarcinoma cell for use in the experiment that I discussed in the article. Here's our list of materials. So we have teratocarcinoma afflicted mice, a 5 milliliter syringe, a centrifuge, trypsin, Hanks balanced salt solution, a freezer, a filter, capillary tubing, and a microscope. You probably are unfamiliar with a few of these, like centrifuge, trypsin, Hanks balanced salt solution, and capillary tubing. But don't worry, I'll explain all of these as the process goes on, what they are and how they were used in isolating the teratocarcinoma cell. So, to begin the experiment, scientists took a bunch of mice that were already afflicted with the teratocarcinoma and let the teratocarcinoma tumors inside of the mice culture and fester. They took a 5 milliliter syringe to extract the tumor and cancerous fluid, specifically targeting those embryoid bodies that we talked about in the article, if you remember those. They then put those samples into a vial to be repeatedly centrifuged. And so what a centrifuge is essentially is just this device with a platform that spins really fast. And the purpose of it, here's the platform first with, let's just put the one vial in here. And so the purpose of the centrifuge is to spin the vial so quickly it creates a vortex with centrifugal force that separates the contents of the vial based on density. So what that essentially ends up doing is, if you'll see here, once it goes in, it starts off as just a solution where you can't really tell the difference between the solids and the liquids. But when it comes out after being centrifuged and being subjected to the centrifugal force, you'll see that because it separates based on density, the fluid stays on top while the solid pools at the bottom. And so this is what the scientists use to isolate the teratocarcinoma cells from the surrounding fluid. After a few runs of the centrifuge, scientists transferred the sample to a dish in which they input both trypsin and Hanks balanced salt solution. So trypsin is a type of biological molecule called an enzyme, right? And so what trypsin does specifically is it breaks bonds either between molecules or between molecules and other surfaces. So what trypsin is doing here specifically is breaking the bonds between the proteins in the teratocarcinoma cells and the walls of the actual dish itself to further isolate it. And Hanks balanced salt solution is just a patented solution to make the cells last longer, keep them healthy, and in a state ready to experiment on. So after allowing the culture to grow inside the dish for 24 hours and further separate, they put it into a freezer to be repeatedly frozen and thawed to kill off any bacteria. And once that bacteria was killed off, they put it through another syringe with what you call a millipore filter, which is this attachment here in the blue, to get rid of any debris that might have gotten into the sample throughout the experiment, to make sure that it was just the teratocarcinoma cells that they were looking at and had in the experiment. So finally, after all of that separation, the scientists were sure that they had just the teratocarcinoma, and so once they put it back into the dish, they used what's called capillary tubing, which is just a really thin tube used to extract small amounts of a substance. So say just this amount of substance for the stratocarcinoma, right? And they manipulated the medium to make sure that it was just one cell that was extracting into the capillary tube. And to verify that it was just one cell that they were extracting, they took the capillary tube and put it under a microscope. So imagine these are the walls of the capillary tube intensely magnified by the microscope. And what the scientists were looking for as proof of isolation was this one dot. And that one dot was the singular teratocarcinoma cell that they needed isolated for the experiment. Okay, so I'm just going to give a quick little, really brief run through of the entire process one more time, just because I know it's a little bit confusing. So first, they took mice infected with the cancer and extracted the cancer and used a centrifuge to separate the fluid from the solid, making sure that only the cells pooled at the bottom. After that, they took that vial and put it into a petri dish where they put in trypsin and Hanks balance salt solution to make sure that the cell could grow for 24 hours, but stay isolated and healthy for further experimentation. And they put that solution, they put that medium into the freezer to kill off any bacteria, and put that into the filter to get rid of any extra debris that might have gotten into the sample, which they then finally extracted using capillary tubes and then examined under a microscope. So hopefully that all made sense. I tried to make it as clear as possible with the different colors. But yeah, that's just the process they used in making sure they had a single cell to be able to test the pluripotency and validity of every single teratocarcinoma cell to make sure that they were all pluripotent. This study done by Kleinsmith and Pierce ended up being revolutionary in both the fields of stem cell research and just cell culturing in general. This method of cell isolation is very widely applicable and has been used countless times by other scientists in different professions. But for stem cell research specifically, this was key in understanding how carcinoma, cancer, and pluripotent cells divided, and helped determine the direction of future research and the validity of certain theories, such as the stem cell theory of cancer. So this Kleinsmith and Pierce experiment really was a complete landmark in the scientific community.